Hi all, Dr. Clark here again. Today in lab we're going to do the very first part of the bacteria lab. And so what I'm going to do is I'm going to inoculate some Petri dishes, excuse me, some Petri dishes, and see what's growing in a given environment. So we can do some calculations, some index, to see what environment has more diversity of bacteria. Now, just because it has a more diverse bacteria doesn't necessarily mean that it's dirtier or worse for you. Okay? Now, the more bacteria you have in an environment, the greater the chance is that you'll have a bacteria there that could be harmful to an individual. Okay, so we're going to look at a few different environments, and I'm going to demo out how this is done. Okay? So first, let's just talk about a Petri dish. So this is a Petri dish, okay, and it has two main parts. Okay, pretty simple. It has a lid, okay, which you can see here, okay, and then it has a base, okay, which you can see here. Now, in that base is a nutrient auger, okay? And that nutrient auger is going to have the nutrients like nitrogen and phosphorus and potentially a little bit of carbohydrates that is going to be needed for bacteria to grow. Okay? Well, our first plate that we're going to do is an exposed plate, and that's just going to be exposed to the native atmosphere, the native air in a region. This is much like Louis Pasteur's work when he was trying to figure out do all cells come from pre-existing cells? You know, that third tenet to the cell theory. This is kind of the same way that Louis Pasteur did his work, except for he used a nutrient broth, which means that no auger was added, so it didn't solidify. Instead, it was a liquid. But it's the same premise. Okay, so what I'm going to do is take the top off of this, and I'm actually going to write what time I took it off at and today's date, okay, on the edge of this, so I then know that the date and the date and the time, and then I'm also going to write that this is an exposed plate. And I'm just going to leave it sit there on the table, open, for however long it takes me to do this lab. Okay? And that'll be about an hour to complete all the tasks in the lab, and that should give me a fairly good amount of time for that to collect bacteria and fungi and other things that are just floating around in the atmosphere in a lab. Now, when I'm done with that, I'm going to put the lid on it, and then I'll write the time that it concluded at. Okay? So the next dish, and for most of you, you're actually going to do two of these dishes, okay? And that would be a dirty finger, clean finger dish. And so what I want to do first is to the dish, I want to just draw a semi-straight line, and I say semi because it's not straight, down the middle of the dish. You can draw that with a ruler to be better than me. That's pretty easy. Again, so straight line down the dish. Now to one side, which I am going to label dirty, I'm going to take my index and middle finger, and I'm just going to rub them down that side, okay, just like that. Now that's dirty. Okay. I'm going to close that because now I'm going to wash my hands with some material. Okay. In this case, I'm just going to wash it with soft soap, Fresh Breeze, hand soap. Okay. This is an antibacterial soap, okay, hand soap. So I'm just going to wash my hands. Most of you have seen somebody wash their hands before. so this is probably not all that interesting. But I'm just going to wash my hands, for a little bit of time. I'll probably do it for 1 minute or so. And make sure that I'm kind of, washing all the parts of my hands, not just concentrating on washing, the 2 fingers that I scrubbed onto the nutrient dish. But I'm going to wash it. And so I said a minute, but really 30 seconds is plenty. Okay. I'm going to dry them. Okay. And then I'm going to, on the other side of the dish, I'm going to wipe the same 2 fingers down the other side of the dish. Now, like I said before, you guys are going to do this with 2 dishes. So you can either look at males versus females. Okay, so I'm going to remove it with that hand. So this is still clean. I haven't touched anything with these 2 fingers. Then I'm going to run it down the other side. Okay, now I'm going to write clean on this side and note that I used hand soap to clean it with. Okay? And so you're going to do this twice. So maybe you want to compare males versus females. Or maybe you want to compare a couple different substances, a couple different types of soap. So maybe you want to use hand soap and then someone wants to use Perel. Or maybe you want to use dish soap and hand soap. Or maybe you want to use bleach, like a 10% bleach, or maybe an alcohol or something like that. Something that you would potentially use, I guess. Not very many people wash their hands with bleach, but, something that you would potentially use to wash your hands. Then you can test, what's the ability for that substance to remove bacteria, to kill bacteria, to dislodge bacteria. Maybe you want to do it with both people use hand soap, but one person uses a brush for 30 seconds. These kind of things, okay? So that's really up to you. And if you're just watching this and you're going to be using the data that I collect to answer the questions, I will use a hand brush and soap, but I'm not going to do it again today because I've already washed my hands. And so I want to collect the bacteria, the exact same kind of situation that I did before. Collect the bacteria, normal activities and things like that between washings, and then I'll wash it again using a scrub brush and hand soap this time to see if that's going to dislodge more bacteria or change the diversity of the bacteria. Okay, so that plate is done. Okay, there will be another one with it. The next one is I'm going to choose 2 environments to swab. Okay, and then I'm going to inoculate those dishes. So I'm going to use 2 sterile cotton swabs. Okay. So I'm going to open the cotton swab, and what I want to do is I need to get this cotton swab wet. Okay. So what I'm going to do is I'm going to use just a little bit of regular sink water to get the cotton swab wet, and then maybe I'm interested in swabbing a certain object. So this is wet, and now I'm just going to swab, maybe I'm going to swab a light switch. Okay, so this is a light switch, so I'm going to swab the top of the light switch, rolling the cotton swab around, and I'm going to swab the inside, outside, and bottom of this light switch. Okay. Now I have bacteria from that light switch on this cotton swab. I have the Petri dish right here. So I'm going to open it up, and then what I'm going to do is I'm just going to twist that and spin that around in the center of the dish, making sure that I got all the sides collected and good to go. When that's done, I'm going to toss that in the garbage can, put this lid on, and now we're going to inoculate this dish. Okay? So hopefully you can see this. What I'm going to do is I'm going to fire up the Bunsen burner, and then I'm going to do what I call a modified continuous streak. Okay? So I'll show you what I'm talking about in just a second. So first, I'm going to light that on fire, get the Bunsen burner going. Now, when you're flaming, this is an inoculation loop. Hopefully you can see that. There's a loop at the end. Okay? And when you're flaming a loop, you need to concentrate on the kind of light blue flame, on that burner. Okay, so right at the top of that light blue flame is going to be the hottest portion of that Bunsen burner, of that flame. And then you want to flame it all the way to the edge, making sure that it's glowing red the whole way. Okay? After that, you then can open your dish up. Okay, so you can see me do this. I'm going to open the dish up, and then I'm going to make sure that it's cool so I can just touch it to the edge over here, away from the bacteria that I smeared in the middle. And then I'm just going to run the inoculation loop from the middle down to one edge. Okay. After that, I'm going to reflame the loop. Okay. So the loop's been reflamed. And then I rotated the plate. I rotated the plate 180 degrees. So I'm going to make sure it's cool. So I'm going to touch it outside on the edge there. Go to the center. I'm going to grab that bacteria that's still in the center, and I'm going to move it to this edge. So that's what I call a modified continuous inoculation. That's been inoculated. So now I'm just going to put the cover on right on here, okay, on the edge of the plate. So note that I'm writing on the edge where the nutrient auger is. Okay, so the bottom of the dish, because the top, the lid, can spin. So you don't want to really write on the lid of a Petri dish. And you don't really want to write on the bottom of the dish, because what will happen is you're going to be looking at bacteria on here, and any color or anything that you write on the bottom will make it harder to distinguish what's inside. So I'm just going to write here, light switch, and that gives me an idea that is what I swabbed. Okay. All right. I'm going to do one more off camera, just to save, your guys' sanity. So I'm going to choose one more thing to inoculate. Then I'll come back and I'll show you how to do the throat and the nose. You might want to fast forward it, because I'm actually going to stick a cotton swab up my nose, which is gross, and in the back of my throat, which I'll make some gross faces, as most people probably will. So if you don't want to see me do that, you can fast forward it to the end, or play it at a faster speed, so you don't have to watch the gross details. Alright, so I'll bring it back and I'll show you again how to inoculate one more time, and then I'll show you that kind of throat and nose swab with that inoculation going on. Okay? Okay, I'm back. I already selected another sample for the environment. I selected the door handle to the lab. So we'll do a comparison of door handle versus light switch, see which one houses more bacteria. Now I'm going to do a nose sample for myself, and then I'll do a throat sample for myself. So I'm going to do a nose sample, and then right after I'm going to do a throat sample, and then I'll inoculate both of them at the same time, just so I don't have to have the Bunsen burner running constantly, because it's kind of loud and wastes gas also. Alright, so to begin with, I am going to wet one of these cotton swabs, and then the key to a good nasal sample is you need to go, pretty deep into the nose, and so you need to go up into the nasal cavity, not just on the ridge of your nose, so you're not just sampling kind of dust and things like that. You want to sample things, bacteria profiles and maybe fungi that are deep up inside the nostril. So I'm going to stick it up in there, going to rotate it around. It's kind of hard to talk. Okay. And then just like the other samples, I'm going to smear that material inside the center. Oh. Sorry. I have to wipe my nose. Alright. I'm going to put the lid on that. Okay. And we'll come back and inoculate that real quick. Okay. The next one, I'm going to do what, you really want to put water on these. It's much more comfortable to do that, and it picks up a lot more bacteria. So cotton swab wet, now I'm going to the back of my throat. You're going to need to swab it just like you would get a strep test done. So the back of your throat, go up and down a couple times. Again, if you gag really bad or if you get bloody noses and things like that, choose someone else if you have lab partners to do this for you, or do it on someone else. Do it on a kid, husband, wife, whatever, someone that can take it, okay? Oh, that was not fun. I don't recommend doing that too often. Alright, so, got it. Now, again, I'm going to flame the loop and inoculate this material. Okay, so flame it. Again, you can just test to see if it's cool by touching it to the edge. Okay. And then go ahead and start in the center. Move it to one edge. Flame that loop again. Rotate that plate 180 degrees. Okay. Touch it to the edge. Okay, it's cool. Streak it from the center. Okay, and that one's done. Okay, flame the loop again. Notice how when I flame the loop, I am not, to get this to cool down faster, I'm not shaking it in the air, I'm not blowing on it or anything like that, okay? The key is to let it air cool without introducing a bunch of bacteria. So I touch it to the edge, go from the center, move it around, flip the plate 180, burn off any excess bacteria. It's flipped 180. Okay, I'm going to touch it to the edge. Okay, it's cool. Go from the center, move it out. Okay, reflame the loop before I put it back so I'm not introducing bacteria to the container that it was in. And good to go. Okay. Alright, so with that, all my dishes are done except for, again, like I said, I'm going to have to do 2 finger dishes, but I have to do them at separate times because I only have 2 hands, and I don't want to have one, mess with the results of the other dish. Okay? So it's been roughly 45 minutes, so I can close my exposed dish. Okay? And these will all go into the incubator. And they'll go into the incubator at 37 degrees Celsius. And I'll incubate them for probably 2 days. That's normally pretty standard, at least for this kind of bacteria work. 2 days, and then we will pull them out and look at them. Now, if you're doing this at home, like you collected dishes from me, I gave them to you, or someone else gave them to you, again, your temperature is not probably going to be around 37 degrees Celsius. It'll probably be closer to 20 or 22, about 70 degrees Fahrenheit. So you'll want to stick those in kind of a cabinet region, or somewhere that's protected and warm, and you want to leave them in there for a maximum of 5 days. After 5 days, the nutrient agar will start to dry out, and it'll be very, very difficult to analyze any bacteria. So I highly suggest put them up there, check on them every couple days, and see what you get. Now when the bacteria is grown, and I'll show you, then we'll come back and I'll do lab number 8, which is bacteria number 2, where I'll show you how to count the bacteria, and I'll also show you how to go ahead and find out the actual shape of the different bacteria, whether it's cocci, bacillus, spirillum, and whether or not it's gram-positive or gram-negative. Okay? And so that'll be next time. Once this bacteria is grown out, then we can analyze the diversity of the bacteria and how to really kind of classify bacteria in a couple ways. You can classify them as a colony, so the shape and size and texture of the colony, color of the colony, but you can also classify bacteria by their genus or genera, and that's the actual shape of the bacteria. So you have to pull the colony off, and you have to do some microscope work and some staining work to get at that. Okay? So next time, that's what we'll do. I'll also, tomorrow, I'll do my own second-hand one where I'm going to use a scrub brush. I'm not going to do a video on that, but I will show you the results from those. Okay? Until next time.